പിന്നെ നമ്മുടെ വീഡിയോ
Yes, the 8th class students, welcome to the meet. So we are discussing about chapter 2, microorganisms, friends and foe. So in this we are discussing about bacteria. So we are discussing about bacteria. In the previous class we have discussed about habitat of bacteria. Habitat means where do they live and the size of the bacteria. What is the largest bacteria, smallest bacteria, so that we have discussed. And also we have discussed the shapes of the bacteria. Based on that we have many divisions: focus, spirale, vibrio, etc. like that. So now we are going to discuss about structure and function. We are going to discuss about the structure and function of bacteria. Okay, structure and function of bacteria. Okay, mainly bacteria contains cell wall. Okay, the bacteria contains cell wall. Bacteria is a prokaryote. We already discussed, right, in the previous class. So it contains what? Cell wall. What does it contain? Cell wall. And it is made up of several layers. Several layers. It is made up of several layers. These several layers is made up of peptidoglycan. 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 So it is otherwise known as mucopeptide. 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 So you all members know cell wall is the outermost layer of the plant cells. You all members know that. So the outer layer which is present in the cell, it is known as cell membrane. But in animal cells, it is the cell membrane. So here, what is the outer layer of bacteria? Cell wall. Okay, the cell wall is made up of many layers. That many layers it is known as peptidoglycan. It is made up of peptidoglycan, which is known as mucopeptide. Mucopeptide. And okay, next, if it contains, okay, this cell wall if it contains, okay, cell wall, this is the bacteria. So this is the cell wall. It's made up of many layers. Okay, many layers. Just I'm saying, just I'm showing the typical bacteria. We are discussing about typical bacteria. So this is what cell wall. The cell wall is made up of several layers, which is known as peptidoglycan. And if this cell wall contains, if this cell wall contains teichoic acid, teichoic acid. If it contains teichoic acid, then that is known as gram-positive bacteria. Gram-positive bacteria. If the teichoic acid is absent in the cell wall, what is present? Teichoic acid. If the teichoic acid is absent, then that is known as gram-negative bacteria. Gram-negative bacteria. The bacteria, both gram-positive and gram-negative bacteria, both are made up of cell walls only. But what is the difference? Presence of teichoic acid. If teichoic acid is present, gram-positive. If teichoic acid is absent, gram-negative. So what is the outer layer of the bacteria? Cell wall. Outer layer of the bacteria, what is that? Cell wall. Cell wall is made up of several layers in the bacteria, both in gram-positive and gram-negative bacteria. Next. And these bacteria contain several structures. Several structures for attachment. This bacteria will attach to other organisms with help of some attachment structures on their outside body. This is generally flagella or cilia, which helps for the locomotion. You know, in unicellular organism or in microorganism, we see outermost structures which helps for locomotion and feeding. Pseudopodia, cilia, flagella, isn't it? Like that. So this bacteria is also contains the outer structures which helps for the locomotion. which helps for the locomotion. What are the outer structures which help for the attachments? Let us discuss. One is glycocalyx. Glycocalyx. First one is glycocalyx. Outside of the body attachment. And another one is glycocalyx. And another one is flagella. Flagella. These are the structures present in the bacteria. First structure is cell wall. Outermost structure we are discussing. Outermost structure it is made up of cell walls. In some bacteria, picoic acid is present. Then we call it as gram-positive. In some bacteria, picoic acid is not present. Then we call them as gram-negative bacteria. And another parts are present. What are those? Glycocalyx, flagella, and pili or fimbriae. We call it as pili or fimbriae. And another one is what? Sex pillar. To produce what this sex pillar? You know all members. Sex pillar is for producing young ones. Okay. These four structures are for what? Attachment. For what these are for? Attachment of bacteria. Attachment of bacteria outside of the cell wall. These are present. Okay, please note down. These all are outside structures. Outside. Which are present outside. What are present outside? One is cell wall. It is made up of several layers. That several layers together it is known as peptidoglycan mucopeptide. And in the cell wall, picoic acid is present or absent. If present, we call that bacteria gram-positive. If it is not present, we call it as gram-negative. And along with the cell wall, other structures are present outside. What are those? Glycocalyx, flagella, pili or fimbriae, sex pillar. What are these four meant for? Attachment of bacteria. Attachment of bacteria. So here, next we are discussing about another structures. This is the outside structure. After cell wall, what are present? Let us discuss. Next. Next, what is present? Cell membrane. After that, cell membrane. The inside structure of cell wall is the cell membrane. Outside structure is the cell wall. And now what is that? Cell membrane. So it is the lipoprotein issues. Lipoprotein. It is made up of lipids and proteins. Lipoprotein means what? It is made up of both lipids and proteins. What is the function of cell membrane? It helps for the binary fission. Binary fission. Binary fission means splitting into two. Splitting into two means bacteria undergo binary fission for in the reproduction. Splitting into two. One bacteria will give you two. One bacteria will give two. So what is the cell membrane? It is made up of lipids and proteins. Lipoprotein. So what is the use of cell membrane? Function of cell membrane? It helps for the binary fission. And after cell membrane inside, what is present means cytoplasm. You all members know cytoplasm is the jelly-like substance. You have studied, okay, in the what is that in the lesson? What is that? Cytoplasm. Cytoplasm is the jelly-like substance present inside any cell. So in this cytoplasm, what they contain? They contain ribosomes. 70 ribosomes are present. In the cytoplasm, what are present? 70 ribosomes are present. These ribosomes are made up of 2 units. 50 So ribosome structure will be like this. So what are present inside the cytoplasm? Only ribosomes are present. What are ribosomes means? They produce proteins. Okay. Ribosomes will produce proteins. You will be studying in other classes. What is the use of ribosomes? In each and every plant cell and animal cell, ribosomes are present. So in bacteria cytoplasm, what are present? Ribosomes are present. Which type of ribosome? 70S ribosome. It is made up of two subunits. This is 70S ribosome. One is 30S and one is 50S ribosomes. Okay. And these ribosomes, they are present in groups. These ribosomes present in groups. Polysomes or polyribosomes. And along with the ribosomes, water is also present. Can you see the board? 70 to 85% of water is also present in the cytoplasm. Once again, I am repeating the structure and function. Both structure and function we are discussing about bacteria. So bacteria structure is made up of outer layer is the cell wall. We have discussed already. Okay, it is made up of several layers, mucopeptide. And that several layers contain stearic acid, positive, if not present, negative. And glycocalyx, pili, sex pili, and fimbriae. Okay, flagella, these are present on the cell wall only. And after cell wall, inside what are present? Cell membrane is present. Cell membrane is made up of lipids and protein, lipoprotein. And what is the function of cell membrane? It helps for the binary fission. Means bacteria should reproduce more bacteria, no? At that time, the bacteria will divide into two. So at that time, cell membrane will actively participate. And inside the cell membrane, what is present? Cytoplasm. In cytoplasm means it is jelly-like substance. In the cytoplasm, what are present? Ribosomes are present and water are present. Ribosomes and water. Which type of ribosome is present? 70S ribosome present. 70S ribosome is made up of two units. One is 30S and another one is 50S. 30S and 50S. Generally, we have two types of ribosomes? 70S ribosome, 80S ribosome. In bacteria, which type of ribosome present? 70S ribosome. 70S ribosomes are made up of 30S and 50S. If they are present in groups, then that group we call it as polysomes. That group is known as polysomes or polyribosomes. And how much water is present? 70 to 85% of water. Okay, this is about cytoplasm. Next one, nucleus. Generally, you all know what is that? If we draw a typical cell, if we mention a typical cell, what are present in a cell membrane, cytoplasm, nucleus, we will study. Form of structure, we study about cell membrane. And after that, we study about cytoplasm. In the cytoplasm, what is present? Nucleus. But in the bacteria, what is present? So let us write down here these 2 points. Then we will go with the third point. Write down in your notes. So if you, whatever I have written on the board, if you write in the notes, no need of writing from the material. Okay, the material just used for learning. This is for writing only important points. Please write. Yes, listen here, students. 70S ribosomes. I have said 70S ribosomes and marked it as 2 subunits 30 and 50. 30 plus 50 comes up to 80. But that is not the thing. 70S ribosomes are made up of 2 units when they meet together. 2 units when they are formed by sedimentation, the remaining 10S will go off. Okay, that is the reason. Don't think it as wrong. 30 and 50. Correct one. Next we will discuss about another point which is present inside. So you all members know that cell it contains what? A nucleus and as well as what is the nucleus is present and cell cytoplasm is present. Okay. So but in the bacteria, nucleus is not covered by a nuclear membrane. Nuclear membrane absent. If we cover water, for example, water is there. Okay, water is there. If you pour in a water bottle and if you close, it have a definite shape. But once you think if bottle is broken, what happens to the water? They will float, no particular shape. Like that, nucleus is not covered by a nuclear membrane. Okay, that is why we call it as nucleoid. So means what? Nucleus is not covered by nuclear membrane. That is the thing. Nucleus is not covered by a nuclear membrane. Generally, inside the nucleus, we see chromosomes are present. What are present? Chromosomes. Nucleus is the main part of any cell. Why? Because nucleus only will control all the functions. All the functions, whether to do or not to do. Okay. In the nucleus only, genetic material is present. What are present? Chromosomes are present. This is the nucleus. What is that we can say? Nucleus is not definite. Not definite. What is that? Nucleus is not definite. Means what? Nucleus is not covered by nuclear membrane. And only single chromosome is present. Single chromosome is present. And DNA is circular. Circular DNA is present. But it is double stranded. Double stranded means like this. Double stranded means like this. Circular, no? So circular means what? Like this. Circular mode. This is the circular DNA. Circular DNA. Circular DNA is present. This is actual DNA in our bodies, but in bacteria, this type of human. This is human DNA. This is human DNA. I'm saying the change between human DNA and circular DNA. The actual structure of DNA is double stranded, 2 strands, spirally. Okay, spirally present 2 strands over one another. But in bacteria, DNA is circular. Next, histone proteins. Histone proteins absent. Histone proteins absent. Next, in the nucleoid, plasmids are also present. Plasmids are also present, which are autonomous. Autonomous. Autonomous means self. Autonomous means self. They can produce selfly by their own way. So, plasmids in bacteria, there are 3 types. F plasmid, R plasmid, C plasmid. F plasmid, R plasmid, C plasmid. F for fertility. This F plasmid is for fertility. And R for producing genes. And the C plasmid for killing others. These are all present in nucleus. What is that? First one is what? Nucleoid contains single chromosome. DNA is circular. Histone proteins absent. Plasmids are present. And 3 types of plasmids are present. This is about nucleus of bacteria. Once again, I am repeating. Listen carefully. So, each and every living organism comprises of what? Nucleus. Okay. If we call it as a living cell, then it definitely contains nucleus. Okay. Once again, I am repeating. If we are saying any cell as living, bacteria is a unicellular. We are multicellular. In our body also, each cell contains nucleus. But here, in the bacteria also, it is a living cell. It is unicellular. So we are calling living means what? Living means what? It is undergoing many functions daily, within a minute, within a second, like that. So what should present? Nucleus. But why we are calling it as nucleoid means it is not covered by a nuclear membrane. The difference between nucleus and nucleus. In our body, what is present? Nucleus. In bacteria, nucleoid. Why we are saying nucleoid means it is not covered by nuclear membrane. So nucleus is not definite. And in the nucleus, what is present? Only one chromosome is present. In the nucleus, what are the structures present in the nucleus? One is what? One nucleus, one chromosome, single chromosome. And another one is what? Circular DNA. What is the DNA? Circular DNA. This is the structure of circular DNA. And another what? Histone proteins absent. Generally in the nucleus, what should present? Histone proteins present. In our body nucleus, histone proteins are present. But in bacteria, what about histone proteins? Absent. And what are present again? Plasmids. What are present? Plasmids. Autonomous means they can form. They only will produce another plasmid. So plasmids are, how many types of plasmids are present in bacteria? 3 types. F, R, C. What are the 3 types? F, R, C. What is the function of these all 3 means? F for fertility. Means if one bacteria should produce another bacteria, F plasmid is necessary. And the genes, transformation of heredity from one bacteria to another. That is for R plasmid. And the C plasmid for what? Attacking or killing other bacteria. For food. Okay, for anything, for shelter, they will attack other bacteria. So which organ will help? Which part of the nucleus will help? C. C plasmid will help for killing other bacteria. So this is about nucleoid. This is the structure and function of bacteria. Structure and function of bacteria. First one is cell wall, second one protoplasm, third was nucleus. Cell wall, protoplasm, nucleus. Cell wall is made up of several layers: teichoic acid, gram positive, gram negative, glycogen, pili, next pili, fimbriae, flagella, sex pili. Next cell membrane, it is made up of lipo, protein. Next cell membrane is helpful for binary fission. Cytoplasm, it contains what? Ribosomes and what is that? Ribosomes and what? And after that, what is present? Nucleus. Nucleus, what are present? Nucleus is not definite, single chromosome, circular DNA, histone proteins absent. 3 types of plasmids are present: F for fertility, R for genes, C for killing others. We have learned structure and as well as function of the what is that? Bacteria. Okay, write down and we will go for nutrition. Next topic is nutrition of bacteria. Next, the nutrition of bacteria. So structure and function it is over. How they will get food? Nutrition nothing but getting nutrients. Means getting food. So nutrition in bacteria. Nutrition in bacteria. First one is photoautotrophs. The bacteria are photoautotrophs. Means autotrophs means they prepare self. Auto means what? They self prepare food. Photo means what? Light. Photo means what? Light. Generally, photo means autotrophs. They will prepare food with the help of light. Here also they will prepare food with the help of light. So, what are they? Let us write. Examples. Examples. Purple sulfur bacteria. B slash means bacteria. Okay. Purple sulfur bacteria. Next. Chlorobacteria. Chlorobacteria. These are the examples. Another one is green sulfur bacteria. So I am keeping B on the top of the slide means it is the abbreviation form of bacteria. Don't write like me B and slash. So bacteria. Okay. Next one type. Next one. It is photoheterotrophs. Photoheterotrophs means photoheterotrophs. Photo means light. Okay. Heterotrophs means they depend on others for food. Means they take the food and eat. They take light as energy. What is the difference between photoautotrophs, photoheterotrophs? Means here these bacteria will take light and prepare food. But without preparation of food, they take directly solar energy as food. No preparing food. Direct they will take what is that light? Means what? Here heterotrophs means depending. Photo means light. This group of bacteria, what they are doing? They are trapping solar energy and producing food. Okay. But here these heterotrophs, photoheterotrophs, what they are doing? They depending on solar energy. Directly they will eat. They won't change energy to food. They directly taking energy. Okay. What are the examples of that bacteria? Let us write. Rhodospirillum. Rhodospirillum. Purple non-sulfur bacteria. Okay, this is the difference between photoautotrophs and photoheterotrophs. Next one. Okay. How they are, these groups are based on how they are getting food. How they are getting food. One group of bacteria are trapping solar energy, preparing food and then getting nutrients. One group of bacteria, they are taking directly from energy into the food. No change of, they are not undergoing photosynthesis. These are photosynthetic. These are not photosynthetic. They can trap solar energy, but no preparation of food material, no photosynthesis. And another type, third type. Next one is chemoautotrophs. Chemoautotrophs. Autotrophs means chemo, they will take carbon. Autotrophs means they prepare food material. Photosynthesis. They undergo photosynthesis. How they are undergoing photosynthesis? By taking carbon. By taking carbon. What are the examples? Let us write. Carbon. They use carbon during photosynthesis. Hydrogenomonas. Examples. Hydrogenomonas, Stylobacillus, iron bacteria. These are the examples. And another one is chemoheterotrophs. Chemoheterotrophs. Chemo means carbon or chemicals, and heterotrophs means they are depending on chemicals. Okay, so what are those? Parasites and saprophytes. If they are depending on others, we call it the saprophytes. Okay, parasites. So what are the examples? Means bacillus. Bacillus means what? Rod-shaped bacteria. Salmonella. So what is it? Heterotrophs means they are depending on others for food. Which food they are taking? Chemicals or carbon? Chemicals or carbon. Their heterotrophs. What is that? Photoheterotrophs means what? They are depending on others as sunlight energy.